Hello, everyone. We'll be starting in a few minutes. Thank you for joining today's meeting on Machine Learning 1 course. Stay tuned and we'll be starting in a couple of minutes. Thank you. It's 8 p.m. and I think it's time. So let's start. I think we have a few participants. Many of you would be joining us soon. By that time, let's go through something of the introduction. So, hello, I'm Mohit Mazumdar, and welcome again to Introduction to Bioinformatics. So, today we are going to review work, revise what we did in the last class, and then we are going to learn new things together. But before we go into the learning, it's very important to look at the progress. So we will be looking at your progress. We have been looking at your progress every week, and we have been seeing good trends. So okay, so that we can make out that each of you are learning, right? That's the reason we are looking at it. Maybe at different pace. Maybe some of you are learning at a different pace. Maybe some of you are learning at a higher pace, something like that. But important thing is to start and be consistent. So the correlation is pretty simple here. The more time you spend on the platform, the more you will learn. And if you face any issues, we are well connected to you via forum. These sessions, we have a group in WhatsApp, we have Facebook, LinkedIn, etc. So let's just quickly go to the organization page and find out the progress. So let's go here. So we are on 9th, next class would be on the 9th September. We are on 6th September and we are finishing Machine Learning 1 today. The Machine Learning course will finish today. So as you see, many people are learning PCA, which is good because we covered it in the last session. So let's go into the progress. So in the forum, we've been pasting the links for the YouTube videos. So all the programs, all the sessions, all the workshops that we are doing are being recorded and put in the forum for you guys, so that when you finish, maybe when you finish this course, or maybe when you go back, and if you have a confusion or if you have a query, you can refer to the video and see that query to be resolved. So maybe at times, what happens is that many parameters, we forget about many of the parameters that are there in the pipelines. So yeah, that's how it is. So I mean, these are good. So this week has been good. So Shreyashi again has been using a lot of server. Ayushman, Shubhrajit, Soham, Hindal. So these are the people who've been like, there are a few people I see, they've been constantly, using the server. But I'm now more interested, to see, I mean, how we have progressed. We started around on 19th of July. Uh, I think that's that's where we started, and then wait we started on 19th of July, apply, and then we went till this is 6th of September. Okay. So this will give us the entire, the entire spectrum, like the way you guys have been, using the server, using the education platform, and what you guys have been up to, who's doing what, and how many points you guys have collected, how many courses you guys have done. Because in the next phase, like we are finishing our course here, the major courses. In the next class onwards, we'll be talking about the projects, and we'll be, slowly transitioning into the projects. So it's very important to understand that how much we have learned so far, because all of the things that we have learned will be applied in the project. So if we don't know, it would be really difficult for you guys to do the project. So anyone who is interested in the project should finish a few of the courses, at least Introduction to Bioinformatics, Transcriptomics 1 to Transcriptomics 3. And if you're interested in genomics, then Introduction to Genomics and Genomics 1. And if you're interested in metagenomics, then Metagenomics, Introduction to Metagenomics and Metagenomics 1. So those are the courses which we have gone through during this sessions, and we expect that many of you should be able to finish those courses. So I see Sridhar at the top of the point schemes. Ayushman is also there. Shantani Ghosh is there. Shantani Haldar, Shubhrajit. Many people are there. Shreyashi, many of you have got more than 10,000 points. So these top 4 people have got more than 10,000 points, which is very nice. But I also expect many of you who are here around 5,000 points to reach to 10,000 points. So here I see many people have started using and then maybe lost interest or have any problems, or maybe not having getting time to finish the courses or run the pipeline. So I would really recommend you guys, because you guys have now enrolled into the course and we are having these sessions. So it would be really nice if you guys can also finish the courses, and then we can think about the project. So many of you I see have just started, and I don't know what happened. So I mean, we are already here, we are always here to help you out. If you have any problem, then come to us, talk to us, if you're not being able to finish the course or run the pipelines or something like that. So I see a lot of progress. A very good amount of people have done a fair amount of educational and server practice. But I think now it's time for many of you to also do the same, and so that we are on the same page, and we will be able to do a nice project after this. So yeah, I will go back to the presentation now. So I mean, the overall progress, if you see here, it's nice, but again, I mean, some of you should again be using our material. So back to the presentation. So we did looked at your progress, and then we talked about that we'll be talking about the projects in the next session, where we'll be discussing about the opportunities to work with us on different aspects of multiomics data analysis. And you can also we can also work collaboratively with the faculties and MIT who are interested in doing one of these projects and these ideas that you have. So please also talk to your faculties, and if they're interested in doing a project, then we can do a project together, like a group project. That would be very nice, because then it will be more translational. Okay, so next we will go into the review. So in the last class, we talked about the data set a lot, and supervised machine learning methods, meaning that we started with some exploratory data analysis and data mining. And now we are heading towards completing the whole data analysis of this high throughput data. So after the preliminary analysis, now the real question is, how do we get some results that we can then interpret? And by interpretation, of course, now we are looking for annotation of genes. Yes. So as I said, we were looking into the data set. So we looked into this publication with multiomics data and started our analyses using transcriptomics gene expression data. We looked at the 7,000 genes. So all these genes that we have in our table of expression, when we looked at those samples, they represent 4 different subtypes of breast cancer, right? We have normal like, we have basal, we have Claudin low, we have luminal. So we have 4 subtypes of breast cancer. So after talking about the data sets, we started to understand the methods. So the methods, machine learning methods, right? So machine learning methods to make sense of this heap of data. That's why we started learning about machine learning methods. So the machine learning algorithms are nothing but statistical and mathematical models. That's the reality, right? The models are popular and working well nowadays because we are recording all kind of data, and data has pattern, right? Data has pattern. Like we are looking at different type of collection of images, your chat logs. Maybe there is a pattern to everything. Like every individual has a pattern, right? and and we are learning about these patterns. So these patterns are helping us in making accurate predictions. That's why these patterns are important, right? We started with supervised machine learning techniques because the data, we started with a data with which has no labels. So a data with no labels, a data with which about which we don't know much, is we use supervised machine learning techniques on such kind of a data, because we are looking for patterns within that data. So we are first identifying patterns. So that is the supervised machine learning method. Sorry, unsupervised machine learning method. I always keep on saying supervised, so I'm sorry about that. So in supervised machine learning method, we know about the pattern. So we have already recorded a pattern. So what we are trying to do here is that we know about the pattern, and now we are trying to predict the patterns that we are that are the new patterns. So anything new which comes, a new thing coming in, so we want to predict it based on already recorded pattern. So that is a supervised machine learning method. So we discussed in the last class mostly about the unsupervised machine learning techniques, which we are reviewing right now. So we talked about data mining. So for the first step, so the first step towards data exploration started with data visualization. So data visualization, why did we require visualization? So we are trying to solve the curse of dimensionality. So we talked a lot about dimensionality, that we are working with a data which is multidimensional. So the data, dimensionality has to be reduced to make any sense out of it, right? So we looked into PCA. We performed PCA, right? So we talked about this multi-dimensional space and reducing the dimensionality, right? If you see the dimensionality of this space, if it is a two-dimensional image, what you can see here, if it is a really flat image, a two-dimensional image does not give us enough information, right? We looked into that. But when we twist the data set, like here it is moving, right? And you can see different angles. So when we twist the data set into three-dimensional space, and really that is what actually PCA kind of does, right? It kind of finds these components that maximize variance in our whole data and plot these projections closest to what is the original data. So it is not the genes, but it is the PC projections, which somehow correlates with the variation that we have in the gene expression data, the variation that we want to capture, sort of that. So we are trying to visualize this here. This is the first step, right? So we can see that as that cube twists, the way we understand the data structure changes. So how many groups are there? That is what something we are looking at. Where those groups are located, something we are looking at. How these groups are, compared to one to the other, etc. These are the things that we are actually looking at. So we can really try to find the best angle, and once we find the best angle, we captured that in a PC. So yeah, that was PCA, right? Also, we practiced the PCA on a small data set. We performed PCA on a large data set and on a small data set. The large data set had almost 7,000 genes, and the small data set had 15 genes in our case. So I'm sorry, here, this is an old slide, I guess. So we had 15 genes that in the 700 genes plot, we were able to classify the genes into two separate groups, right? And then we saw that with a smaller set of genes, in our case 15, it showed us very good separation. It showed us very good separation into four different groups. So we understand that the variability in the data, as you see here in 7,000 genes, the variability is high, right? And in 35 genes, the variability is somewhat, I would say, is less random. So the variability in the data affects the performance of the algorithm, right? You can totally make that out. And also that a clean and well-defined data set with samples from each group representing variety amongst themselves, right, help us with better classification of data, because there are different kind of patterns recorded for one kind of subtype, so that it understands the pattern. So that's what I'm coming at. We also discussed genes and samples, and in this talk we will talk more about features. In our example data set, the features are the genes that are aiding in better separation of the groups. Okay, so the features are the gene. We are looking at genes that help us, that has been helping us in separation of these groups, so that we can say that, okay, beyond this expression, this is this subtype, beyond this expression, this could be this subtype, something like that, right? Okay, so after doing PCA, so yeah, Claudine low, basal and normal and luminal. That's how the separation was, which we saw in the last class. So after doing the PCA, we did H clustering, right? And in H clustering, we looked at the lineage and why we used, a depth of 4 cluster count, why we used 4 clusters, because we thought that it's a 4 groups. The distance was Euclidean because it's the kind of line. So there are different kind of lines, obviously. Transpose we used because we are looking at the samples, things like that. So we ran this pipeline, and what we were looking at a dendrogram, right? So we got these results from hierarchical clustering based on our selection of the depth, right? We can see different number of groups, and so you can see here it's 2 clusters if you cut it from here, way up. Okay. Now, if you cut it from here, you get 4 clusters. So what I mean is that you get a lot more definitions as you move down. And one of the things that we discovered talking about this is that it actually makes biological sense. So how? So what I mean is this classification makes biological sense. I will tell you how. Sorry. So, these types of subtypes, these classes are really defined, these breast cancer subtypes. I mean, they are really defined by long history of trying to understand how breast cancer. So in our study, as we saw last time and discussed, where all these samples came from, how to categorize this type of disease. And the first approach was based on histology. So based on the source tissue, based on the staining for individual protein that were present or absent in the sample. And eventually what next generation sequencing gave us in terms of the genes, that it helped to separate accurately the number into the number of groups, right? Here samples are clustered into well defined groups and of 4 or 5 different subtypes. That is what we are looking at, right? So biologically, how biologically? So biologically today we kind of separate this luminal into luminal A and luminal B. So as a result, we are actually looking at five different subtypes or classes. So that's an important observation, and I think within those subtypes it is also important to understand that we expect to have outliers. Okay. So even here in this picture, if you see the sample HCC 202, HCC 202 is a sort of an outlier. It is highest from everyone else, as you see, and it's constantly coming up in other analyses as well. Okay, so let's summarize what we have observed so far. So we have defined groups, okay, that we know about, and we know about those groups. The four subtypes. Then we have biology that tells us otherwise, that there may be really 5 different groups, right? And then we have individual specification of samples that might make it outlier, like in this case. This is what we saw. And so it's like what I'm coming at is like, it's not always, a model does not always explains everything. That is what I'm coming at. So there are exceptions to the rule. So that is the conclusion from our supervised machine learning analysis from exploring this data. So today we are going into classification, perhaps. So supervised analysis talks about two approaches. One of the approach is classification, and another one is regression. So those two are really used for prediction. So classification specifically can be used for feature selection, and that is another aspect that we are going to look today. How do we use these methods to predict? How do we use these methods to understand classes? How do we use this method is something that we are going to learn today. So we are going into the supervised machine learning methods. Before we go into the supervised machine learning methods and I talk about the things, do we have any questions? Does someone have any doubts till now? So I'll take like I'll give it a pause. Okay. So supervised machine learning. Supervised machine learning takes in data that has to be labeled, right? Typically, the labeling is done by us, like humans. So in biomedical projects, this data could be clinical data, could be image data, or could be other phenotypic data. So this data is used to train a model. So the method varies. So as the principle for different algorithms that are involved in supervised machine learning methods. So there are different algorithms that does the supervised machine learning methods. I mean, that do supervised machine learning. But how they do, that varies from method to method. For example, Linear discrimination analysis, linear discriminant analysis, LDA. LDA attempts to draw a separation line between the center of the classes in the training data. So that is one type of machine learning method. The method is, it is essentially learn from the differences between various type of classes. So here, if you see, there's a source data. So the source data has different type of shapes, right? And we record these shapes by labeling them. So this is green by color. So we are labeling them by color. We are separating them by color. So this is green, this is red, and cyan, and then different colors, yellow. So we are building a training data set with the known labels. So we are saying that, okay, this is this and this is this, and we are telling that classifier about the training data set. So here we use different algorithms like LDA, random forest, and then we are trying to predict new data. So when a shape comes like this, it does predict it correctly to be in a specific class. But when you get a shape like this, like a star, what would you do? So that star would be a, for, I mean, it will not be able to predict it. So because it does not know the pattern, it is not, it has never been trained on that pattern. So new data can be assigned, but that data has to fit, the model. So in this case, the star does not fit. So if you put input like stars, this will actually reduce the accuracy of the model, right? So I'm seeing a few chats. Anub, is skitlearn? Oh, this is beyond this class, so we are not going to discuss this. So yeah, going into the data set, right? Yeah, we're looking at the data set. Yes. So I think you guys have received the link to the data. This is the link. This is a tiny URL link. So let me show you guys about how the data looks first, right? So I will just copy this link and open this in a browser, so that you guys can also, if have not downloaded, you guys can go on and download the data, and so that we can maybe run the analysis together. And if not, then you guys can always uh, repeat this once you see me doing it, and then always and then of course you have the video as well available with you, so that you guys can see and repeat whatever I am doing today. So I have downloaded two sets of data files. One is supervised 15 genes, and these two files, so this is dot something, this is a mac file. So for supervised, we have two data set, train and test, for 7,000 genes, and similarly for 15 genes we have a train and test. So we have given you a data set for 7,000 genes and a data set for 15 genes. So just look at the data. Let's look at the data. So supervised 15 genes. Let's look at 7,000 genes first. So train data set. So let's open this via Excel. I'll just open the Excel. So yeah, thank you, Shubhrajit, for pasting the link. So whosoever does not have it can download the files, and of course you guys can go ahead and do it together. Yeah, these are very simple, so don't worry, it will not take you much. So let's look at the train data first. So the train data is something labeled data, right? We talked about this. So we are telling the machine about the label data, that okay, this is how the data should be. If this is the so can you guys see my screen? I should ask first, because if sometimes what happens is, like, I'm doing it here, and because of the internet, sometimes what happens is that the screen loads, kind of slowly to many of the participants, and then sometimes they miss out. So I think if you guys can see my screen, so this is your training data set. Just look at the chat. Sure. Okay, so this is the training data set. So this is the gene names, the ensemble IDs, which we discussed earlier. And this is the class, and this is the ID. So what is the ID? ID is what we already know from the data. So these are the samples from which the data is coming from. And these are the gene expressions from of different genes in those samples, right? So here we already know about the class of that sample. So we do know about that this is a luminal sample. We do know about that this is a Claudin low sample, and henceforth. So we are telling that to the machine that if you see a pattern like this in different genes, if you see a pattern like this, if you see a pattern like this, if you see a pattern like this, and if I show you just plot, you'll understand that these patterns are actually different from each other. So if you see, I mean, yeah, they are almost and that is the reason because they falls under the one category of luminal. So yeah, so as you see, like there's a pattern with every class. So that is the pattern we want to capture. So this is your training data set, right? And similarly, we will now open the test data set. So this is the test data set. Let's open this up as well. So here you will see that we are not providing it with what? We are not providing it with uh, the class. So we are something that is something that we are predicting. What we are predicting? We are predicting the class, that which class this genes will fall under, or which class with which will help, which genes will help us to classify the class. So that is what we are looking at. So these are the two data sets. I think you guys have understood the meaning of these data sets. If you haven't, please unmute your mic and ask me question if you have any queries regarding this data set, because it's going to be important for you as well when you do your own thing. Then maybe you have to that would be a different story altogether. So I'm surprised that no one of you have asked anything yet.
Sir, AD had raised his hand. Maybe he has a query.
Yeah, I mean, if you write in chat and if you ask, unmute yourself and ask me, then it would be rather helpful. I cannot see you guys raising your hand. So please ask, unmute yourself and ask me the question. Okay. Yeah.
Hello, sir.
Yeah. Please.
Sir, am I audible to you?
Yeah, you're audible.
Yeah, sir. Can you please explain the differences between this train and the test data set? Means, how did you prepare this train data set? Because in the previous class, what we have done about this unsupervised part, we had a data set with the marked one and the unmarked one. So, is there any difference between the marked, unmarked, and train versus train test? Sorry.
Okay, that's a good question, and it's a genuine question. So the marked and unmarked in supervised machine learning was only one data set, right? That's the one complete data set, one file, one data set, whether it is marked, whether it is unmarked. So the reason to mark it was to see the results, that whether uh, these groups that we have are from a same subtype or not. So in unsupervised method, we actually don't know in a real life situation, you actually don't know whether that is actually a basal or luminal or normal like or Claudian low. So but we have marked it just to show you guys that the clustering is correct, because it is falling, all of them are falling into their respective categories. But in supervised machine learning method, what we are doing, that all that 7,000 data set that we took, the unmarked 7,000 data set that we took, or a marked 7,000 data set that we took, we broke it into a train data set and a test data set. One file, there was one file, we broke it into two files. Okay, so why we did that? Because we are trying to make the classifier, or make the supervised machine learning method learn about the data first and then predict about the data. So there we were looking for patterns first. So we understood that there is a pattern, we understood that this is basal, this is how it looks like. So this is the pattern of basal, right? We got a group, right? And if we take those genes which comes in basal group, that will define that group basal, right?
Yes,
So we took that information and put it in the unsupervised method, where we told the machine that this is basal, this is how basal look, this is normal like, this is how normal like looks. So we build a training data set like that, with some number of samples, and then we build a test data set to test the classifier, that whether you are predicting the right values or not. So it's like testing it. Once you have train it. So it's like you go prepare for an exam is like a training, right? And when you go and give the exam in your MIT university or whichever university, that's the test. So in the training time, you open books and you do all the notes and you learn.
yes,
And when you go to the exam, you don't take help from all those things. There you r- write what you have learned. So it is exactly like that. So first you so that you learn about the patterns, and then you predict whether you know the patterns right or not. So there is only one data set which we have divided into two, one to train it and other to test it. Did you get it?
yes, sir. Thank you, sir. And one more question, that you said that in the luminal type, you will further differentiate between luminal A and luminal B. So,
Mm-hmm.
how do you understand from the dendrogram pattern itself, or was it a hypothesis? Means, how did you get to know that there will be two furthermore subtypes inside the luminal one?
So in biology there are two more subtypes. So from biology I know,
Okay.
from the experiments I know that there are two subtypes. Okay. So in the luminal, so here if you see that there are multiple levels of clustering happening. Okay. So what is happening is different clusters have different I mean they are going into different group. So here we have 8 clusters, but in reality if you see here 8 clusters, so this is one group, this is one group, this is one group, this is one group. So one, 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 one. So four, four, 8 groups. So here we are dividing them or classifying them into 8 groups. And I have said, based on the information that I know, that we might have 5 groups here. So that is something which is not clear. That's a problem that I said in supervised machine learning, that this sort of information is what you get. Okay, so this is more like a hypothesis driven approach. Many times you are learning about these new things, about a new subtype, or something that is coming experimentally, and then you correlating with this data. Okay, now I understand why this is grouping is like that, because this is a different family, or this family has a different function, or something at a level of depth that makes them differ from each other, right?
Okay, sir.
Did I answer your question?
Yes, yes, sir. Thank you.
So one more question, Mohit, people have, that the train data set which is being prepared, is it a random process or not?
No, it could be a random process. So trained data set is a if it could be it could be a random process only when it is done by an algorithm. But the preparation of train and test data set is one of the biggest challenge, right? This is one of the challenge in machine learning, data mining, or classification. That how good your train and test data set, that will make the result or the classification and its accuracy and other parameters will depend on it, that how well you have defined the train and the test data set. So the train data set should have the variations. It should have captured the variation coming from one subtype. So what do I mean is, in theory, that see, if we are classifying, for example, apple and mangoes, right? So we classify apple and mangoes using their shape, right? So if there is a case where apple is like looks like more like the shape is coming up as a mango, so that would be a false prediction, right? Something like that. So similarly, how well you define those parameters, how well you define your training data set, is something which will influence your results. So how do we do it? It's based on your experience, and the most important part is to understand your own data. So once you understand your own data, then you will be able to separate them properly into test and train data set. So how do you do that? I mean, it is something like something of an experience, that with which comes with an experience, right? So if you say that I am taking this class, I am doing machine learning and introduction to machine learning 1, and I will be able to do this and learn everything, that is literally like impossible. That is seriously impossible. What we are trying to do here is to make you understand and give you an overview of things, how they work, but it would require you to go in depth and learn more. And I think what we are trying to do here is a collaboration. So it's like a research based learning. So it will take time for you to get to a research level of this sort, where we are learning about the algorithms, the train, how to make train and test data set. That would come when you keep on doing projects and do hands-on practice a lot with the server and henceforth. So did I answer your question?
Yeah, thank you, Mohit.
Okay, so we have a lot to cover. I am just at the database. We have not yet, went into the demonstration. So let me quickly go into the next step. So we did talked about the data set, and I showed you the data sets and how they look. So next we will go into something that would help us to classify the data into different groups, like decision trees. The easiest way to think about this is, if I can select a gene, if I can select a gene, sorry, where the pattern of expression is so clear that you can actually use a simple threshold and say, okay, based on this threshold just like here, I can classify more genes into two different groups, right? This is just a yes or no, if it's like a condition, right? Decision trees are really a nice way to start talking about classification, because they're kind of simple, right? You can totally, I mean, get it from here, that it is easy to interpret. And basically, if we have to separate everything into a series of rules, we would say, okay, if something equals to something, and if something is equal to something, and something is less than this, and something is more than this, then you put it in one category. And if something is not matching, then you put it in a different category. So here what we are trying to do is, we are trying to put a threshold between them. So out of this data set, this is a result. So okay, so we, out of a data set, we have identified two genes here. based on one gene, if this is the value, if this gene is coming from this, then it goes into this. So I'll give you an example which will maybe help you understand. So first let's run a decision tree for 15 genes, and then we will understand. Let's go to the platform and run the pipeline. So I guess you guys have all logged in, and under the supervised analysis, we will go inside the we will first upload the files. So here, add files. So my files are in downloads, I think. Yes, here. So we are putting 15 genes data, train and test. So select both and then open. So you are downloading here two groups, two data sets into one. Okay. So once you have uploaded the data, there is a button called group selection, right? We are going to select it into two different groups. So group selection. Here, if you see, this is the test and this is the train. So I will put the test into the test one, and I'll put the train into the train one. Okay, I will continue now. Now, what will I do? I will start. Okay. And what we are trying to do is, here you can see there are different number of algorithms. So we started with decision trees. LDA, QDA, stepwise LDA, random forest. Okay, so there are no parameters. Just save it and then just end this. Okay, so this is 15 genes. What we are trying to do here is a decision tree for 15 genes, and I saved it and I run the pipeline. Simple as that. So yes, so I will just maybe wait for the result, or maybe show you the results in some time. Okay, so decision tree 15 genes. You saw how I did it. It's fairly simple. So I've already, I think I've run this one. So let's look at the results. So you get a zip file called 15 this zip file. So you download the zip file, and now you open the zip file here. So I extracted the zip file. So I got the results here. This is of the plot, and this is the prediction statistics. Just open and quickly see what it is. So it is telling me that the overall accuracy of the training data set is 0.92. This is really good number, because the prediction accuracy is 92%. Very high. And this is happening because we are using a set that is a smaller data set, and which we saw that in that data set, all the separations between different groups are well defined. So that's the reason. So let's look at this plot. So this is the plot. So we have got two genes. One is 6299, and another is 9888. So basal, so if you see, based on this threshold, it is class between Claudin low and basal. And here, based on this threshold, it is classifying it into luminal. So we are, based on this data, it is actually classifying groups into three different classes, right? So let's go back to the presentation and let me explain you what we actually have obtained this result. So this is the result you got, right? And these are the two genes. One is 6299. So I took it from the expression table and just plot it. And this is the pattern we actually saw last time as well in the previous class, if you remember. So in this example, there are two genes out of 15 that can help classify three cell types based on expression level of two genes, right? So gene 6299 and gene 9888 in combination can distinguish whether a sample belongs to the cell type basal, luminal, or Claudin low. So here you can see that this gene we started in the beginning talk about the same gene, that a pattern that we are talking about. So if it is more than 4.25, assign it to this group, and if it is less than 5.2, assign it to these groups. Okay, so assign it to either of these groups. Okay, so that is the real point of classification with decision trees. So what is the problem with the decision trees? Okay, there are several problems with decision trees. One of the algorithm that works with in a decision tree has a random initialization. So what do I mean by random initialization? Random initialization means that the first tree will start from some place and we don't know what place. So we will try to find the optimal genes that are separating the data set into whatever classes by doing this random initialization. So one tree starts from a point, and that's what based on that we get this result. So what is happening here is that you get one gene at a time. That is one problem, or two gene at a time, or maybe three gene at a time. But it is not really enough for our need of interpretation, right? We are looking at a pattern. So for our need of interpretation and the question of accuracy and stability, so those are the biggest problems with decision tree, that the decision tree's accuracy and stability is highly questionable. So because it is a single run approach, so how do we solve this problem? How do we solve the problem of decision tree? That what is the solution? The solution is random forest. So if we have a lot of random these random tree formation, so in decision tree we started with one tree. Okay, so beginning point for running, if we have many trees, we might get different result, right? And we also get some different genes all the time. So some results we will get one gene, in some results we'll get another gene. So one iteration after another iteration, the whole idea is to actually develop an algorithm that does this actually on decision trees. So that is why this is random forest of decision trees. That is the random forest. It actually runs multiple decision trees, so 500 so forth. So you can see right here, if we have an instance and a random initialization, the run 1 2 3 like there are multiple runs, and that there could be thousands and thousands of trees. And we see that for a single tree, there could be a single number. I mean, one number can be repeated. So one initialization happened with one gene. It does not mean that another next time the initialization will not start with that gene. So there could be 4 times 5 times initialization could happen from that gene. So what it does is, in this way, it helps in the prediction. How? Because we take voting. So we do voting. Okay, how many times for this specific sample did we actually get this result? You're getting my point, right? If you're repeating it like that, we are seeing that for how many times this result is replicating. So if it is coming up more and more time, probably that's the correct answer. So that is the voting based method with random forest compared to the decision trees, right? Okay. I mean, in case here, if you say we are looking at single tree decision boundary, and this is multiple run. So if you see here, the accuracy has improved while using the random forest. It tells you the number of features that separates this whole data into number of classes, right? So what actually it is doing, the random forest is giving you number of genes, it is giving you number of classes that will help to that will help you to separate the groups into different classes. So it will give you the important number of genes. Those genes will help classify the data into a correct manner. So that is the purpose of running the uh, random forest. So we will go, and now we'll demonstrate and we'll see how it actually works, right? So as I said, if you have if you get a number of genes, and those genes will be able to accurately represent the whole data set. That is something that we are looking at. So let's go and quickly do it on the over the server. Run a random forest pipeline. So I will create another. So let's see if that run is finished or not. Yeah, this run is finished. I have already shown you the files. This is the file that you're supposed to download, and then I extracted this file, and then I got this plot and this statistics. So those are the important values I should be looking at. So next I would go into the random forest. So random forest is what? Random forest is nothing but like multiple runs of decision tree, right? So we have different types of random forests here. Random in the platform, random forest multi-run, random forest multi-run iterative. So one thing with random forest is that because as you see that there are many initialization, and there are number of trees that we can actually go on and on. So it's a computationally expensive method. So it takes a while, to run the random forest for a bigger sample. So it also depends on the size of the sample, of course. So you have multi-run approaches. So you can run group of random forest again, and this is again over that multiple run. You can have more. So these will be helpful in finding something to a point of really accurate, maybe a small data set, but you're looking for something like very accurate study. So you'll do that. So we'll start with random forest. So yeah, but before we have to upload the data, right? And I showed you that we have had the files. So here I will what I will do is I will use the 7,000 genes, right? This time I will use the 7,000 genes and see how random forest is giving us the list of important genes. So I remember Shreyashi did ask me this question, that sir, how do you come from 7,000 genes to 15 genes? So Shreyashi, this is one of the answer. So how do you come from a big data set to a small data set that helps you separate the classes into into groups? So what you saw in decision tree before this, decision tree gives you one gene or two gene. So those one or two genes could be your biomarker, right? Because that one gene is classifying them into different groups. And in random forest, you're getting a signature, right? You're getting my point. So you're getting a group of genes that are being able to classify. I hope so. So let's do the group selection. And this is the test. I'll put it in the test, and this is the train. I'll put it in the train. This is simple drag and drop. There's no hassle. And I'll start. Okay, so what I'm trying to do is to go to random forest. Okay, number of trees 500. 500 is a little too much. I think I should do 50, because I'm using 7,000 genes and you guys are here and we are towards the end. So should be fast. So random forest, 7,000 genes. If you see, like, running these pipelines are really simple. These methods are very complex methods, but this is the beauty of the platform. I mean, these it simplifies difficult methods so easily that we don't have to worry about anything but to do the analysis. That's more important than anything else, because you should learn about the data and the output and things like that. So random forest of 7,000 genes. So I have started this run. So if you see here, so in random forest, you're getting, I said, a group of genes. So these are the two lines. The one suggests the accuracy, and one suggests the stability. So the basic idea is that we have some separation between our samples, and it will be based on some kind of a gene, right? And if we do random forest, then many trees, we will get an idea of several genes that are that are responsible for separation, right? That is what I've told you. So random forest feature importance plot. What is this feature importance plot? This is what, these are the list of gene, these are the list of gene that are, important for the accuracy of the prediction. If you take them out of the calculation, it will increase the accuracy of this prediction. So these genes are very important. They are helping the classifier, to be, to the point that these are like critical. If these are out, then the classification is poor, because these are important genes. I hope you get my point. And here, simply, these are also the set of genes which are important in terms of stability. So if I take one of these top gene, the stability of the predictions are going to be low. So this is a way to come back to a group of genes which are sort of important. The genes that you see are the ensemble IDs. Pratim, okay, thank you, Pratim. We are trying to simplify things here for you, but please follow transcriptomics 3. Yeah, I mean, many of these things are transcriptomics in transcriptomics 3, and always, as I said, you can come back and always ask us if you have any questions. And again, I will say that we are going to have another workshop with you, where we will be again covering many of these things. So don't get worried, and don't get, that this is something that is too much or it's very difficult or blah blah. It will not be, because we'll be helping you guys, and it's a long-term collaboration. As I said, it will happen over time. So this is the idea. You guys have now some uh, you guys have some understanding of the method that is being widely used, the method called random forest and decision trees. So obviously you can go into more depth and understand, the confusion matrix and things like that, which will give you a more in-depth understanding of how the algorithm works. But obviously those are beyond the capability to cater in this small time that we have. So completing machine learning and all the aspects in 2 hours is literally very difficult task. So, but we are trying to give you an overview. So in the next method that we are trying to see is would be LDA. So we've been talking about LDA. So I think it would be nice if we can cover LDA and also see what is actually how LDA works.
So there's a question, Mohit, in the chat. What do you really mean by stability? Can I just quickly say about it, or we should
No,
put
I
it
mean,
later?
really, I can talk about it a bit. That stability is in terms of is a matrix which we calculate based on the classifier's performance. So stability in terms of the prediction, in terms of like how this prediction is making the whole groups. I mean, the classifying the subsets, the classification of the subsets. That is what. So if you take it one of these genes out, the performance of the classifier decreases, of course. And the stability, I mean, the stability which justifies that how how well the perform. It's another performance matrix. So yeah, obviously we can come back to you and tell you in more detail in the next class, which will clarify more of your doubts. We can do that. Maybe in the next class we will address this in more depth. Okay. So next we would go into the linear discriminant analysis. So discriminant analysis, or LDA, the idea is that we want to define, we want to define how to discriminate between several types of samples. This is what we are trying to do here, right? In some sense, LDA, or linear discriminant analysis, is the supervised alternative of PCA. Why? Because instead of finding the best principal components that separates all the samples based on the variance, here we are finding some line that can accurately separate our classes. So here we are looking at a line, and here we are also looking at the centers of these two classes, and there can be separated based on those based on the line that we draw. So there are many types of variation of LDA. So there is a linear you see there are of same shape, and if you see quadratic, you will see different shapes. So all of this is based on different assumption, based on the distribution of the data. So it depends on the distribution of the data, that what kind of LDA you will be running, whether it is a linear or quadratic. So in quadratic, as you see, there are different shapes. Now these are the details that are important to understand, because you have to understand your data a little bit to be able to select the right method. These things, these machine learning methods are different from each other, because their algorithm or the basis are different. So one way is to understand your data and then run the method. Another way, that is the learner's way, is to do by trial and error and see what works. So let's go quickly and run a LDA and see what we can actually get from the LDA. So we do get a graph which actually looks like PCs. There is a lot of similarity between LDA and PCs of PCA. So let's just upload the data for LDA. So we'll do with the small set of genes, maybe, or maybe we can do it with the 7,000 genes as well. Yeah. So the process of data upload is the same. It is the same for every time you run the pipeline, the supervised pipeline. So you have to select the groups, and then you put test into test, and you put train into train, and then you press continue, and then you press start, and then LDA. So here you see there is no parameter. So in LDA, if you could see this plot, LDA produces a number of visual representation of your data that shows how the classes are separated. Okay, so this is a separation of classes, this graph. So let's end this, save, and then LDA 15 genes. No, sorry, 7,000 genes. 7,000 genes. 7,000 genes. Save. Run pipeline. And similarly, we can do it for 15 genes as well, and see the difference, how the separation has changed between the two groups. So I do have the results with me. So LDA for 15 genes. So let's look at the prediction statistics again. Wow. So you see LDA here for 15 genes, 100% prediction. So it's very accurately have classified the basal, claudin, luminal low, and normal like into the groups. And if you see the plot, let's see the plot. Mm, okay, so this is the plot for the 15 genes. If you see the plot, I mean, it's really difficult to, make sense from this. But if you see, I mean, this is the LD1, this is the LD1, and this is the LD2, and this is the LD2, and this is the LD3. So here you can see the representation between LD2 and LD3 is actually perhaps better. Here you can see normal like has been classified, basal has been classified, claudal low and luminal has been classified into respective groups. So this is we knew this because we saw a similar pattern with our PCA as well, right? In the PCA, if you remember, we saw a separation of these groups into this well defined. Here if you compare LD1 and LD2, so here basal and normal like are coming, luminal is here and claudin low is here. So here also the separation is good. But as you saw in the prediction statistics, the prediction is really good. Now, I mean, I wanted you guys to see what will happen when you run LDA with a bigger data set like 7,000 genes, which has many outliers. So let's check out the statistics. Oh, okay. You see here, the statistics have fallen down from 100% prediction. Now the prediction is 86%. As I clearly, size of the data set and variability, the classification accuracy has fallen. So if you look at the plot, so you see here the difference in the classification. You can totally make sense because the classification is all over the place. And I mean, here Claudine low, here also, I mean, this is not so good. So yeah, most of the cases it's difficult, but there is some classification, so it's difficult. What you are seeing here actually is a two-dimensional plot, but it is again an n-dimensional space. So it is a multi-dimensional space. So it works really well if you can visualize like it like that, but that's not possible. So LDA, as I said, gives you is like decision tree in some manner, because it's a single run. So what it is doing is it's discriminating features and trying to rebuild, and it's trying to look at the accuracy, and it's trying to improve the accuracy and things like that. Okay, thank you, Pratim. Discussing about the outliers. So yeah, that was LDA. So I mean, there is an iterative version of LDA, like we saw in the iterative version of decision tree. So the iterative version of LDA is stepwise LDA. So so here we have an like a decision tree, and we saw stepwise LDA. It is in the same sense, but this is useful in comparison to decision tree. LDA, it will select genes that gives the best separation with a little different principle here. The principle of the algorithm here is a little different in LDA or stepwise LDA. So what is happening here? The principle here is called the greedy algorithm. Greedy means that is something related to the greed, that we don't have to really go into the details of greedy algorithm right now, but what I will say that it does select the right genes and give you best separation. So if you see, there is a feature here, parameter called novu. So what it does is if we set the novu uh, more, it works better for if the value is less, it works better for a bigger data set. Actually what we are trying to do here is it is discriminating a lot when the value is low. So it is discriminating a lot of features one after another while doing the runs, iterative runs. And in case if we put it a strict number like 0.01 or 0.1, what will it do? It will keep all the features and then run the calculations. So that is useful when you have a small number of features or small data. We usually keep this value restricted. And if we have a large data, where then we keep on playing with this value with like a higher value. So we will just go and see a demonstration. Okay, quickly we will do this. I'm sorry, we are running out of time, and because actually it's a lot of things to cover. We've been covering multiple algorithms in one class. So we will be just I'm very much thankful to you guys for having all the patience. And we will upload 7,000 genes and see, I mean, how many genes do we get out of this. We saw that in something in random forest does gave us a list of genes. And now we'll see how stepwise LDA does this for us. So test, and the train goes into the train one, continue and start. Okay, so I'm coming to the stepwise LDA. So here I will just make it more bigger because I want to make it test a lot of features, lot of genes in our case. So 7,000 genes, we are doing LDA for 7,000 genes. No, sorry, stepwise LDA. I'm so sorry. LDA 7,000. Okay, save. As I said, it's very simple to run the pipelines actually. So it's gone. So stepwise LDA, where is this? Stepwise LDA for 7,000 genes. Let's look at this data. Okay, so we did the run. So we have a plot, and there I have already discussed the classification. So you guys, I don't think we have to go through it again, but this is a good classification. Let's look at the prediction stats. I'm really excited. Though this is good. See, stepwise LDA gave us a 100% accuracy on 7,000 genes. So this is really good. So feature coefficients. So these are the number of genes. So as you saw in the case of PC1, PC2, PC3, right? You remember those projections? So here also, I mean, this is the LD1, this is the LD2, and henceforth. So these are the genes 1, 2, 3, 4, 5, 6, 7. So stepwise LDA is giving us 7 genes that are very good. I mean that these genes will give us the best separation or a very good separation to further further classification, right? And moreover, I mean, it's a thing like if you can do further analysis on these genes, downstream analysis, and see what exactly these genes actually do. So this is something I think we will cover in the next class, where we will be talking about the biological interpretation. So quickly, as you saw, like SWLDA is also very useful. It gives us important set of genes based on the machine learning technique it uses, right? So let's just go quickly into the SVM, and then we'll be finishing up. So SVM is another method. Support vector machine is designed for efficiency, sorry. So support vector machines are very efficient. Another method that is typically used in this kind of studies are like different kernel based methods. So what we are trying here is that when we construct gene signatures, we talked about gene signatures, right? When you have a group of 15 genes, that's a gene signature. So from a gene signature, we want to develop a good classifier that for predictions, so that we can really scale it up and we can do accurate selections of different classes, and how it is important and blah blah. So a support vector machine is an efficient algorithm that takes the input space. So this is the input space. So you have, different values here at a different input space. So it changes the input space. So it changes the entire feature space. It rebuilds it rebuilds a feature space where the classification is better. So this type of method is very useful when you have different kind of data, when you have multi dimensional data which is very which has different shapes. Like here you see there are different shapes. So what we are trying to do here in PCA, we are actually transforming the whole class to divide it into groups, and this group is divided by a decision boundary. So what is actually happening is a straight line in this case is classifying, it's a line based classification. So we are drawing a line and we are classifying into two different groups. That is what is happening in SVM. But I mean the way it is the way it works is, of course, it's way more complicated. So a straight line separation is the answer, is the trick that we were using, right? So the trick is here is the kernel, right? We have different types of kernels that can adopt different shapes, that can based on those shapes the classification improves. Simple as that. So if you see here, there could be, I don't have that example. So there, here if you see, this is r square and this is r cube. So one is at a linear scale and one is one is at a logarithmic scale. So linear scale would be 1 2 3, logarithmic scale will be 10 100 10,000. So you can really transform this space and into a power of 3. So here we have transformed this space into power of 3, and if we have actually have a separation like this. So if you see, when we moved it like this, there's a better separation. We were able to separate these groups from this group. Here it was difficult because in a 3D space, right? So this transformation happens in SVM. So that is the USP of SVM. So SVM, what it does is, let's say you have two classes. What support vector machine does, it maximizes the distance between the two edges of the class and edges of the here this class and this class. So there's a maximum distance. So once we have a maximum distance, there is a boundary that has been drawn. So in this case, this is a linear kernel because it's a straight line, and it fits the points. So the problem is, when you have a point like here, when you have a point like here, you have a new sample that comes in, and let's say you have a new sample that comes in right here, and that makes your whole data messy, right? So how do we handle that? So it draws a line from that hypothetical line so that it can maximize the separation and gives you a clear classification. So in support vector machine, we are looking for not set of genes, and we are looking for classification, how the genes are being separated. Not like it is not giving us any set of critical genes to do that. So we might have a demonstration of this later. So conclusion, what we did all this while is we did RNA-seq, and then we did a normalization, and then we did test and train. Before that, we did PCA, and we looked into the all the clusters, and then we did multiple. This is ideal way to go about this, from where you can go get different type of genes. So like she asked me that day that how do you get 15 genes. So this is the method. These are the different ways you can go to get different number of genes. So I got 7 genes using SWLDA. I got a lot more genes using, random forest. You do multiple runs, multiple run random forest, you get a different set of genes. So if you can see the results and collate these results, many of genes will be common, and those genes will be very important genes that helps you in the classification, right? So this is what we have learned in machine learning so far. And the best way, I mean, this is something that we recommend. So there is no strict pattern or rules in biological data. That is very, that is very much known, that there is no strict patterns and there is no rules. So the variation is complex, and that is due to numerous factors, of course. So if I say that I know machine learning, and if I only use one method to compare my results, then I would say that is not a good practice. That is not the best practice. The reason for doing multiple methods is using multiple methods is, comparative analysis. First, second, the mathematical algorithm varies from one method to other, that I already explained you guys. Some methods work really well on some data sets, some doesn't. So here a combination of method to understand your data will help you, get better insights, and have, reliable results. So there is a lot of application of machine learning. And I mean, here, I mean, we have, both me and Pratim, who are your tutors, have had an experience in industry and academia working on some of these aspects. So I would like to give the stage to Pratim now, and maybe he will tell you something about these areas which might interest you very much in the future research you would be doing. So Pratim, stage is all
Yeah,
yours.
Mohit. Yes.
Yeah.
Thank you very much. So I think you had a sufficient idea about where to start from and where you would like to go, and understanding about the basic concepts about machine learning, etc. And these are very important. As you know, in the data deluge, there's a lot of data available. So imagine these machine learning methods being so useful when you have a data which is around, let's say, 1 terabyte. You want to classify it, maybe possibly try to understand that where are your classes, what are your classes. So thereafter, this machine learning will be a huge usage to you. So coming back to this slide, the applications that you can see right now in front of you are clinical applications and pharma and biotech applications. By clinical applications, the things that you can see right now in front of you are patient stratification, grouping by the risk, disease classification and diagnostics, and identification of potential responders to therapies, and mechanisms of disease onset and progression. I would request Mohit, if I have the, if he can give the responsibility to share my screen. Mohit, is it possible?
Sure, please, Pratim.
Okay, thank you. Okay, so let me share my screen. I wanted to show you this slide. This slide kind of explains everything. So if you see, sorry. View, and I have a presentation. So see, this is somewhat summarizes whatever you saw, whatever the slide was pointing to. Big data precision and efficacy. It has patient stratification. It has disease classification and diagnostics. It has identification of potential biomarkers and potential responders to therapies, and mechanisms of disease onset and progression as well. So in this experiment, which you can see on the right, we have developed some, we have there, this experiment has taken 1,000 human cancer cell lines. What are cell lines? These are the cell lines. Cell lines are derived from mouse PDX model, which you know all about now. And the number of responders were 11,000 patients out here. And they have tried to stratify the patient. We stratify or classify the patient based on the clinical prevalence during mutations, whatever expressions of those mutations. They can be driver mutations, they can be passenger mutations, and there are also amplifications and deletions. So amplification is overexpression, deletions are underexpression of those genes, and definitely some amount of epigenetic control, which we say promoter hypermethylation. this is a very important aspect of cancer, to be very specific, but because recently we have found, because DNA methylation is a process which silences a DNA from expressing. Now, if the silencing happens to a level which is much greater than normal normal level, the gene becomes most of the genes become silent, and thereby they fail to transcribe into the RNA, and thereby produce the necessary protein that is required in the biological pathway. So we stratify the patients based on this thing, and then we do something called clinically relevant genomic features for sensitivity modeling. This is something, and we test it against the drugs, some amount of, there are 265 compounds, this particular experiment they have done, and they understood, they tried to understand that how are these patients responding to the drugs. So potential responders, and thereby they have identified therapeutic markers. So therapeutic markers are something which are connected to biomarker discovery as well. And as you can see, the drug response modeling. So PTEN is an important gene in the cancer onset and cancer progression. So disease onset and disease progression. So these experiments try to identify whether the PTEN are mutant or PTEN are deleted, and therefore how much sensitive this PTEN mutant or PTEN deleted are to the drug X. And also try to understand that which of these studies, genomics or transcriptomics or many other omic studies, multi-omic studies, how they are really helping us to have more predictive power for predicting mechanisms of disease onset and progression. So see this molecule, as you can see, some of the mutations, these are mutations, these are normal. So just the reverse, in fact. So these are kind of probably mutated varieties. These are non-mutated varieties. These the non-mutated individuals. So non-mutated are resistant to this. So here you can see we stratify the patients, right? And we then try to correlate the in vitro or the in vivo experiment with our computational finding. So this is what, if I can reiterate or recapitulate the process to you, that if you remember the graph that I showed you in the first class, when a particular biologist is thinking all about how is it thinking, he's driven by the hypothesis, he goes back to the lab and does some lot of experiments, starts with an observation, goes back to the clinic, and then all those experiments. But on the contrary, a bioinformatician starts with all the data. So this is the data that a bioinformatician starts with. So these concepts will be very important for you when you'll be doing a project on your own. So as you can see, these are all features. Whatever Mohit talked about, the mutations, features, amplification and deletions, these are features, patterns, all those factor regression, LDA, PCA that you did, these are all gene expressions. So you are trying to classify this gene expression and trying to understand how it is being really helping to stratify the patient, classify the disease and diagnostics, and of course, identifying the potential responders to therapies and mechanisms of disease onset and progression. The applications, if you go back to the slide that is there right now, so I can show you that slide as well, probably. Let me see. Okay, anyway, if you, Mohit, can you reshare your screen, please?
Yeah, Pratim, I think we are also running out of time. So let's
I will just quickly complete and then.
Yeah. Thank you. I'll just share it.
Sure, thank you. I'll just quickly complete it.
Can you see it?
Thank you. Yeah, perfectly all right. So biomarker, you clearly understood that this is a very important thing. For precision medicine, etc., it is very important to identify biomarkers for classifying or stratifying the patient and therefore deducing therapies regarding that. And also, it is very important to detect the toxicity at the in vitro stage. We have to try to understand what kind of side effects a drug might produce. So you can derive predictive modeling. You can derive predictive models out of the responses of the drugs. And a very important part of pharmabiotic is target discovery. What does really this mean? So the term target carries several connotations in the overall context of drug discovery. A remarkable progress in molecular biology has led to the identification of numerous proteins with key roles in the function of both normal and abnormal cells, which has allowed the formation of specific hypotheses and how modulating this function of defined proteins that are linked to disease could be a route to new drugs. So this is a very important concept about identification of target discovery. And to identify target discoveries, machine learning plays a big role. And of course, drug repurposing and repositioning. Drug repurposing is something a very important concept, because many of the drugs which are produced nowadays are, they are meant for something, but they are kind of producing different type of effects. So if you can remember, a strong example would be very interesting to point out. Viagra is a drug which was originally developed for people with a lot of who has heart disease. I'm sorry, there's a lot of background sound might be. I'm sorry for that, but I quickly finish. So drug repurposing is, we will share you some papers. Maybe you can read about it and get back to us with questions. Thank you, Mohit.
So the next class would be on planning a bioinformatics project. So we will be talking about the bioinformatics project that you guys will be involved in. So many of you would be interested in doing the project. Many of you would be interested in finishing the course. So I would recommend everyone to finish the courses first and then jump into the project. So we'll have the next class on Monday, and where we will be discussing about the methods that we talked about today. And also we will be discussing about the projects that you guys will be doing with us and in collaboration with MIT University, of course. So we have this free webinar which we have been talking about to you guys to attend to. I mean, this will be really helpful for you to understand the things that we discussed today. A lot of these things will be happening in more details during this whole course, which is going to start on 16th of September, and there is 20% discount to the students. So I would recommend you guys to take up the course and learn and become a data scientist and data science expert in coming days. So as you know, as I said, that it's going to take some time for you to become a data scientist, as you have realized that there is so much to learn. So we also have a workshop, which I said, which we will be doing on 16th of September. We are already on talking to the your administration at MIT University, and most probably it will happen at the same time, but we don't have a written communication yet. So we'll be telling you guys about the bioinformatics workshop that we will be doing on 16th September, where we will be, mostly defining the projects and how to go about it, for the last 1 month, and, make the most of your learning. So these are the things that we covered today, and these are the things that we're going to cover in the next few days. So I hope that you guys enjoyed today's presentation and are going home with something. I mean, I don't expect you to do or learn everything that we talked about today, because we covered a lot of things. But at least, I mean, you guys can go back, and when you practice by yourself and see the result, and maybe see this video, it would make more sense to you. So thank you so much for all of you because you guys took your time and attended today's meeting and learned something. Hope this was helpful. So I would conclude today's meeting now. Thank you again. Thank you, everyone. So I'm also open to question and answers. We did a lot of Q&A during the chat, and as we know, we are running out of time. But if you guys have any questions, so I see there is a question from Arnab. Excuse me, please. During predicting toxicity responses of a drug in a metabolic pathway, how do I implement ML when there is no clinical data available for a new novel drug lead? This is a very intense good question, Arna, by the way. So I would really love to answer this, but it would take me a while to make you explain and give you the complete answer. So maybe we will discuss this in the next class. So thank you, Arnab. So thank you everyone for joining today. And I would like to thank Pratim and Shubhrojit and Shreyashi and all those people who gave their time. And thank you, Arnab, that you're saying. Yeah.
Mohit, can you speak a little bit about the filling up the feedback form, maybe?
Yeah, I mean, as you know, I mean, the way we have been working here is that we are taking a lot of feedback from you guys, and it is very important for us to learn, to know how the learning has been, because that gives us an opportunity to improve. That also gives us an opportunity for you, gives an opportunity for you guys to express whatever you have learned. So I mean, it's a very good system, and I know we Indians are not used to maybe filling so much of forms and stuff like that, but it's a good practice. So I would really request all of you here to fill up the forms, so that we can have a full learning. So that's a request. So again, thank you everyone, and have a good night and a nice weekend.
Thank you, Mohit, for a wonderful class. Thank you all for joining.
Thank you, Pratham.
Thank you, sir. Thank you, everyone.